Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speaker your question. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speaker, Chris Schultz, PhD, Team Leader, Center for Process Innovation, National Biologics Manufacturing Center.
Well, thank you everyone for joining. Good morning and good afternoon, depending where you're joining us from. My name's Chris. I'm a team leader at CPI in Darlington, UK, and we've been working on a great project with 908 devices. So I'm very happy to be here today to talk you through some of the data. So I'll start off by telling you a little bit about CPI and how we've aligned our functions to this project for 908 devices, and then I'll talk you through some of the data. So we've been using a CHO cell line that expresses a monoclonal antibody. We have it as a platform process at CPI, and we've used some of 908's equipment to characterize and improve the process. So CPI work to de-risk and accelerate concepts and products to the market. And we work at the interface of research and development and the commercial market. We're part public funded and part private funded. We have a lot of UK government assets in the building, which we use to support our partners to de-risk and accelerate their products through from R&D to the market and get them through various technology readiness levels. So we do every end-to-end proof of concepts and scale-up of processes at CPI, with the ultimate goal of reducing risk and reducing costs for our partners and decreasing their time to get their products to market. So in our mammalian process development workflow, which is what we've been using with 908 devices for this project, this is just to give you an overview of the elements of that we use for this project. We do everything from very, very small scale in a shake flask, and we actually go even smaller than that. So 2 ml culture volumes up to ½ liter. And then we've got various bioreactor platforms. So we've got our Amber 15 and our Amber 250 systems, which will look at 15 ml and 250 ml cultures in a bioreactor setting, but we can still do screening with this. And then we can scale up further. We've got 2 liter, 10 liter and 50 liter capacity here. And we've also go all the way up to pilot scale for 200 liter cultures. But for this project, we've focused in on our Amber 250 and our Amber 15 work, and we've also done a 10 liter bioreactor scale with some of the data that we've generated. So our core, this is just to highlight what our core process analysis would entail, and also to highlight one of the devices that 908 have supplied to us that has enhanced this offering. So we would typically in a mammalian process look at viable cell count, total cell count, percentage viability of the cells. In our 10 liter bioreactors, we use capacitance probes to measure biomass for total cell count. We also quantify and look at protein quality. So for quantification, we use our Octet protein A system generally. We can also use our SEDEX BioHT. We will also optimize gassing strategies and look at cell stress through osmolality throughout our process. We would typically look at metabolites, so glucose, glutamine, glutamate, lactate, and ammonia throughout our processes. Oftentimes we would do this daily. We can do this more if required. And we also do cell line development and cell line characterization. So we go all the way from a cell clone to a fully characterized process. One of the devices which I'll introduce later that we've worked with 908 on is the Maven glucose and lactate tracer, which is really a valuable tool that we could add to our, is a very valuable tool to add to any bioprocessing armory. But I'm going to start off by talking to you about the Rebel analyzer, which is a mass spectrometry based product. Essentially, we can look at a wealth of analytes throughout our process. It doesn't require much sample, so we can use 10 microliters or less, so it's very compatible with a small working volume as per the AMBER 15 system that we've used in this project, and it measures all amino acids in the media and also a panel of vitamins, so we get great insights into our process every time. And it's a high throughput system, so you can run up to 96 samples with each run, which is perfect for our 48-way AMBER 15 system. It's great for screening and daily sampling and daily characterization. But if you do want to move up to fewer reactors and react in real time, you can generate one or two data points in around 10 to 15 minutes time, and then react by feeding your culture appropriately. And it's very easy to operate. It's been very user-friendly for us, and we've gained some great insights from using the Rebel Analyzer at CPI. So just to talk you through the experiments that we've ran with 908 devices. First of all, we used the Rebel to characterize a panel of medias with our, just the medias alone before applying them to our CHO process to uncover the content of amino acids in the panel. And then we screened a set of medias. So we adapted ourselves to a selection of these medias and characterized their productivity. And then subsequently, we used the choice media or choice medias from this initial screening to take further characterize the feeding regime. So we used the Rebel to determine the optimal feeding strategy based on depletion and feedback, which I'll show you shortly. And from there, we did further refinement of the strategy using our bioreactor systems AMBER 15 and AMBER 250. And finally, we processed up to scaled up to 10-liter scale, which is relevant for a manufacturing setting. And the goal was to achieve a high titer, control toxic metabolite buildup, and ultimately reduce the requirement for costly chemically defined feeds in the process and achieve the same level of product quality and titer. So I'm not going to dwell on this because I know a lot of attendees will have seen this in previous presentations by my predecessor. But essentially this just was a panel screen. We looked at control medium and a panel of 7 medias, and we just wanted to compare how much of each amino acid was in the media at baseline. And we showed that for this panel of 8 medias, most of the amino acid compositions were very similar, apart from for arginine, aspartic acid, cysteine, proline, and serine. But we had a nice baseline here to then compare to our depletion rates through various runs. So when we adapted the cells to these 8 medias and applied them in an AMBER 15 setting, we measured titer at 14 days. And if we're on the left-hand bar, we can see the control medium expression levels, and we can see that starred here the high performer, medium 6, and the low performer, medium 9, which we looked at in a little bit more detail subsequently. But we can see that we can improve our process immediately by adapting ourselves to different medias. And on the left-hand side so the left-hand panel here shows the amino acid depletion rate in the medium 6, and you can see that the green dashed line indicates 30% of the baseline level of that amino acid in that media. So around day 8, all of the amino acids that we've shown on this graph, and that translates across the panel more broadly as well, hit below 30%, whereas in the media 9, which was low performing, this only happens for the non-essential amino acids, asparagine and serine. But the essential amino acids that are shown on here are not depleting below 30%. So we get an idea of being able to relate productivity of the cells in that media to their ability to take up and utilize amino acids. So this was really useful. And then we subsequently moved on to optimize our feeding strategy. So we wanted to identify the amino acids that were depleted and then refine feedback to hopefully do all of these things. So to increase the product titer, to improve the viability of the producer cell, and to reduce toxic metabolite buildup, including things like lactate and ammonia, and ultimately to see whether we can enhance product quality with a Rebel defined feeding strategy. we used the Rebel to determine which concentrations of specific amino acids were depleted at which points. We calculated the consumption rates based on just the concentrations that were present and were read by the Rebel, and it didn't factor in any chemically defined or CD feed, which I'll refer to several times. We have to use a chemically defined feed in this process to sustain CHO health, but ultimately we wanted to reduce the amount of chemically defined feed and add a defined amino acid feed back in to reduce cost of a chemically defined feed, but also aid cell health. We didn't account for the concentrations of amino acids in the chemically defined feed when calculating this depletion and replacement. But what we aim to do essentially is return the concentration of amino acids to their baseline levels at each feed point. So in this slide, we can see on the left that this is a culture that didn't have amino acids supplemented back in on top of the chemically defined feed, whereas on the right-hand side, we can see our amino acids supplemented back in. So we didn't supplement in serine and asparagine, which are the green and red lines, but all of the other amino acids return almost to 1 times or what we're calling baseline levels. And it's important to note here that these time points are taken 24 hours post feeding. So they will never reach 1 times, but they almost do on each occasion. So we can see that our feeding strategy is restoring our amino acid concentrations back to baseline. And what effect did that have on the titer of the product? So in blue on the left-hand side, we can see normalized product titer in a low chemically defined feed environment. So with 0.5 times the manufacturer's recommended concentration. And in purple, we can see 2 times the manufacturer's recommended concentration of chemically defined feed. And what we see is that when we apply the low level of feed, when we supplement with low or medium amounts of amino acid, which is based on our calculations from the Rebel, we increase the titer relative to no supplementation by about 30% and 20% respectively, whereas we don't see this effect when we add high chemically defined feed, and actually the additional supplementation of amino acids seems to reduce the titer relative to no amino acids being fed. On the right-hand side, we see the lactate profiles from these 2 groups of bioreactors. Our traces for lactate look very similar to this, but for ammonia look very similar to this, but what we're showing here is lactate. What we can see in purple is that the high fed reactors with 2 times chemically defined feed have a lot higher lactate accumulation than with the low chemically defined feed and amino acid supplementation. So we postulated that this might be the reason for the increased titer, and we've achieved our first goal of reducing toxic metabolite buildup using our Rebel defined strategy in conjunction with a lower chemically defined feed level. Just to quickly touch on this, we did some further refinements to the chemically defined feed regime. We tested whether adding at day 2 or day 3 post-inoculation would improve titer, or whether a bi-daily flat, so fed 4% every day, or a bi-daily pyramid fed, which is fed chemically defined based on the cell culture density. So we would add 4%, 4%, 8%, 8%. I'll show this in a bit more detail later, but just playing with the chemically defined feed regime. I've only shown here 2 different groups out of 12 different groups that will run. So in the bar chart, you can see 2 clusters of 4 bars, and in blue and red, we have the flat fed reactors, and in gray and yellow, we have the pyramid fed reactors. So what we see here is that the pyramid fed with 1 group of amino acids, so G1, and another group of amino acids, which is G1 to G3. In both cases, the pyramid fed reactors resulted in a higher titer. So we wanted to investigate this further as we scaled. So here we moved into the AMBER 250, which is a more relevant bioreactor geometry for further scaling and with manufacturing relevance. We designed an experiment in conjunction with Dr. Andrew Hines and Dr. Christopher Montoya of Sartorius. And what we wanted to show was that whether this pyramid feeding regime scaled and whether our rebel feeding scaled well as well from an AMBER 15 setting, which is more a screening tool, to a more bioreactor relevant geometry, and whether serine and arginine non-essential amino acids, which we've seen frequently were depleted in our early studies, were required as additional supplementation on top of the essential amino acids, and whether this would have an effect on the titer. And also just to see whether our feeding strategy scales and whether there were any further refinements to the amino acid feeding strategy that we could apply. I won't dwell on this too long, but this just shows the groups of amino acids that we added in this experiment. We had 20 bioreactors in total, all with different groups of amino acids, so 1, 2, and 3. Group 1 is the essential amino acids that we've been feeding previously with additional arginine and serine. Group 2 is just a group of essential amino acids that we'd identified are frequently depleted. And group 3 is a group that we identified from our AMBER 15 work as being interesting and potentially increasing the titer if we add it. Group 4 didn't receive any additional amino acid supplementation. And on the right-hand side in the table, essentially, what I'm not going to go through all the molarities, but in the respective columns, we've got an addition of amino acids that would add up halfway to baseline, all the way to baseline, and double baseline in 0.51 and 2, respectively. What effect did these different mixes have on the cell density and the viability? So just for simplicity, I've only plotted the pyramid-fed reactors here because when we analyzed the data, it was very clear that the pyramid-fed reactors performed best. So on the top 3 panels, we have the viable cell density, and the bottom 3 panels, we have percent viability, and mixes 1, 2, and 3 respectively as you go through the columns. In mix 1, VCD, relative to control, was fairly comparable. With mix 2, the control VCD the control cells grew faster than the mixed supplemented cells, but this, as we'll show shortly, this didn't have a detrimental effect on the titer. And with mix 3, the addition seemed to decrease the percent viability at the very end of the culture. But 2 things to note here are that for mix 2 and mix 1 in the percent viability, so the bottom left and the middle bottom, we can see that the viability is higher when we add the amino acids in both cases. These set of graphs is just to show 2 examples of amino acids that were included in mix 1 and mix 2. So isoleucine on the top 3 panels and lysine on the bottom 3 panels. And I've marked where the 2 times, 1 times, and 0.5 times concentrated amino acids were added. So we can see really nicely that in the pink lines for the 2x fed in mix 1 and mix 2, that the amino acid increases in both cases. For the 1 times concentrated, it plateaus and stays fairly flat throughout the culture, and in the red, it decreases. So we can see that our rebel-informed strategy is scalable. Another thing that we identified in this is that tryptophan is depleted. Tryptophan is depleted in the 0.5 times chemically defined fed reactors. So tryptophan we didn't include as an additional supplementation in this run, because in the chemically defined feed it was identified, so we didn't think we would need to add it in. But for this run in the AMBER 250, we did notice it was depleted almost to 30% by day 8. So this was something we wanted to investigate at 10-liter scale. And just finally for this experiment, what we see here is the titer. So in blue, the blue horizontal line marks the control titer at day 9, and in purple, the control titer at day 14. What we see is that for mix 1, at 0.5 times concentration and 1 times concentration supplemented, we see an increase in the titer of about 10% in both cases for mix 1. And for mix 2, at the 1 times concentrated supplemented amino acids, we see a similar effect. comparing mix 1 and mix 2, we don't see a great deal of difference when we add in the additional non-essential amino acids. So essentially what we took forward from here is the mix 2 1 times concentrated amino acid supplementation, partly because we didn't need to add the non-essential amino acids to achieve the same titer, and additionally because I haven't shown the data here, but the ammonia accumulation was lower in the mix 2 than the mix 1 supplemented cultures. So for our 10-litre bioreactors, we selected the choice conditions from our AMBER 250, which was the 1 times concentrated essential amino acids, histidine, methionine, phenylalanine, valine, isoleucine, leucine, and lysine. We didn't include threonine because that is in the chemically defined feed that we were adding. And we did a pyramid-fed strategy. This shows here the volume-to-volume proportions of feed that we added on each day. So 2% on days 3 and 5, 4% on days 7 and 9, etc., to show that this correlates with the VCD over that period. And we also analysed whether tryptophan had an effect, which we'd identified from the AMBER 250 as a potential the lack of tryptophan being inhibitory to product production. So we have 3 bioreactors in this setup. Bioreactor 1, we used another 908 device, which I'll introduce you to in a second, to control glucose feeding at 2 grams per litre. And then bioreactor 2, we had 2 bolus-fed regimes where glucose was fed from 2 grams to 4 grams per litre. And bioreactor 1, for the continuous feed, had tryptophan supplemented. Bioreactor 2 did not, and bioreactor 3 did. So what this enabled us to do was to compare bioreactors 1 and 3 for titer to see whether continuous versus bolus feeding would have an effect on titer, and compare bioreactors 2 and 3 to see whether tryptophan would have an effect on product titer. The Maven device has been really exciting to use. It's an enzymatic glucose lactate detector. It's a probe that goes into the bioreactor. It has a membrane inside which allows analytes to diffuse into the probe, and which is then carried off to a sensor in this box. And it also has a PID control which can be attached to a pump and feed glucose in depending on the readings in real time from the Maven. We found that it's very comparable in terms of glucose and lactate from our offline SEDEX readings, which is very accurate. So it's very accurate in terms of its measurement. It's very accurate in terms of its feeding. And because of the way that it works, with analytes diffusing into the probe, no sampling is required, and no loss of bioreactor volume occurs. So it's a very straightforward piece of kit, and it gives a really granular profile of glucose and lactate, as I'll show you shortly. I'm not going to dwell on this too long, but this just shows how the probe works. I've described it verbally on the previous slide, but what happens is you prepare a salt transport buffer which travels down the probe. The pumps pause every 20 minutes. In this case, the Maven can read glucose and lactate every 2 minutes, but for this set of experiments, we did it every 20 minutes. So the pumps will pause and allow analytes to diffuse in, and then the salt buffer carries the analytes into a sensor which detects glucose and lactate. We get very detailed profiles of glucose and lactate using this system. And we successfully employed its PID control function to feed glucose depending on when it reaches the trigger point, which abrogated any fears in our hearts of the reactors running out of glucose. So what do we see in these reactors? Essentially, cell viability is very comparable across the 3 reactors. Viable cell density is also very comparable across the 3 reactors, and pretty high at the end of culture at day 14. This is an example of a Maven trace. What we see is that the spikes that occur throughout this run correspond with the chemically defined feed additions and their proportional size relative to the pyramid feeding, which is very encouraging. And also we can see from between day 4 and 5 that glucose is stabilized and retained at just below 2 grams per litre. So when the trigger point of 2 grams per litre is is detected by the Maven, it starts feeding glucose, which is sustained throughout the run really nicely. If we compare that to one of the bolus fed reactors, this is the minus tryptophan reactor, you can see that essentially the concern will be that you'll get glucose and lactate stress throughout the run, and also we run the risk between day 8 and 9, the culture almost reached a crashing point where glucose was depleted in full. But you can also see the power of the Maven and how accurately it detects glucose and lactate in real time. And the grey line shows the plus tryptophan bolus, and the traces match very similarly to the minus tryptophan bolus. But as I previously alluded to, this point here between day 8 and 9, while we're at home, was a concern, and the Maven abrogates any concerns of such a crash occurring. In terms of the lactate profile, they were highly comparable across the 3 reactors. The main difference to note is that between day 9 and 10, the reactor that didn't receive tryptophan had a small spike in lactate, so the lactate switched back to increasing and accumulating, whereas in the other 2 reactors it continued to decrease. And the reason I've flagged this is because shortly I'll show you a potential effect of this in terms of our product titer. Just to briefly show you this essentially shows that our rebel-informed feeding strategy is working well. We're sustaining. So top left, you can see histidine, for example, lysine and valine. On the left-hand side, you can see that at each feed point, we almost feed back up to baseline levels. So that shows that our amino acid depletion profiles from the AMBER 15 are nicely scaled, and that the reactors are never short of any amino acids, essential amino acids that they may require. Some of the other profiles indicate a slight decreasing trend in feedback, so it suggests that we may be able to refine this strategy even further. So for isoleucine or methionine, for example, we see that we don't always feed back up to baseline levels. So these amino acids are utilized at maybe at slightly different rates, or slightly different rates throughout the culture, depending on the cell density. So this may be further refinable, but we were really pleased to see this. And what effect does this have on the product titer? What we can see at day 7, when there's very, very similar lactate profile, the titer is very similar across the 3 reactors. But at day 9, we can see in red that the titer is lower than the 2 reactors that did receive tryptophan. So in red, the reactors didn't receive tryptophan, and we can see that the titer is lower at day 9 onwards. For this, and this correlates very nicely with the lactate spike that we see. Whether this is directly correlated, we don't know, but what we can say is that those 2 things happened at the same time, and that our feeding strategy and our addition of tryptophan has improved product titer. So in summary, we've really enjoyed working with the Rebel and the Maven. The Rebel's been successfully employed to characterize our CHO mAb production process really well. We've been able to calculate feedstock depletion rates throughout this process, and we've been able to use that to inform an optimal strategy, which is cheaper because we can use less chemically defined feed at great cost, and it can also have a benefit on the toxic metabolite buildup, and without sacrificing titer. And we've also shown that our Rebel process was scaled very nicely through AMBER 15 all the way to 10-liter reactor scale. And then in the last set of experiments, we've shown that the Maven gives very detailed glucose and lactate utilization profiles. We were able to use it to control glucose feeding at several different levels. I haven't shown other levels in this, just to 2 grams per liter, but we have used it to feed at lower levels as well, and controls it really nicely. And we've also identified a metabolic shift that we may not have seen without the Maven in terms of the lactate increase in the tryptophan depleted reactors. And this has all improved our process overall in terms of product titer. So with that, I'd just like to thank the team at CPI and the team at 908 Devices. It's been a pleasure to work on this project. And thank you all for your attention, and I'm happy to take any questions.
Okay, great. Thanks, Chris. And as a reminder to our audience, you can go ahead and type in your questions for our speaker now. So our first question is, this project obviously created a lot of data. Was it easy to analyze the data from the Rebel and Maven whilst running the bioreactors to react to the requirements of your process?
Yes, thank you, Alex. It was actually, I mean, particularly when we were zooming in on fewer conditions. So early in the project, we were looking at 48 conditions in parallel. So it would be difficult in that setting to react to, but there's a tool that's available that 908 have developed, so you can download it. A jump jump software tool essentially. So we just export the files, import them into jump, and then you can toggle between conditions and which amino acids are depleted, which are restored, and you can get a good idea in not quite real time, but probably from the time you've got your sample to the time you've had a chance to have a quick check, probably an hour's time. For this set of experiments, we didn't react in real time in terms of the amino acid feeding because we'd already predefined the conditions in discussions with 908 before going in. But in theory, if you've only got a couple of reactor systems running, you could react to the data in real time. Yes, and it was fairly simple to do so with this tool.
All right, great. And our next question is, are you focusing on essential versus non-essential AA when designing your feed? In many cases, the non-essential AAs deplete faster, but it's not the problem because cells can synthesize them.
Yeah, that's a really good question. I mean, we've looked at we've only really shown a snapshot of the data from the group from the whole project. So we've looked at various groupings of amino acids. What we've mainly found is that the essential amino acids seem to have the biggest impact on, or the depletion rates of the essential amino acids are having a bigger impact on the output. But there's certainly a lot more refinement we could do looking at non-essential amino acids, 100%. I mean, it's both, well, it's a very good thing. The Rebel generates so much data that you can find so many correlations. If you could sit with it for days, or you could sit with it for a few hours. So it's difficult sometimes to not get lost in the amount of data it provides. So for the purpose of this project, we've focused in on feeding back the essential amino acids, but there's certainly some interesting findings we found with including non-essentials in the groupings as well.
And were there any found changes to product quality or sequence variants with AA supplementation? Excellent data and experimentation. Thanks for sharing.
Oh, it's a pleasure. No, thank you for the question. I mean, we've really enjoyed the project. It's been really interesting. In terms of product quality, so we would typically look at charge variant analysis, glycosylation patterns. We mainly looked at this in more detail at AMBER 250 level, and we selected 8 of the 20 reactors to look at. We haven't seen dramatic differences, to be honest. It's more been an impact on the titer that we've seen. We haven't seen huge or really staggering differences in any of the other quality attributes. We haven't done any like stability studies or anything like that. Yeah, I hope that answers your question, but if it doesn't, please reach out to me offline and I'll be happy to clarify that for you.
And what was the titer of the AB strongly related with the biomass? Meaning that with the optimized feeding strategy, do you also get very high cell densities?
It's a great question. I mean, I think it's certainly correlated. In other project work we've done here, we see, of course, a strong correlation of antibody titer with the biomass. In particular, I'm just trying to think back to, for the AMBER 2, I would say generally yes, it is. Generally yes, it is. I mean, we were hoping to be able to see a bit of a more striking impact on cell density with the feed regimes that we've implemented. We didn't see particularly big differences in terms of density. It was more fine-tuning the composition. Towards the end of the set of experiments we did, where we looked at tryptophan, the cell densities were very comparable between the plus minus tryptophan in terms of they had everything the same apart from the tryptophan, and that did increase the titer, having tryptophan supplemented back in. Predominantly, yes, biomass would have the biggest impact. We weren't sure whether we would be able to improve VCD and growth patterns with an amino acid feed specifically. We've mainly seen big impacts on biomass using the chemically defined feed as opposed to the amino acid supplementation, but we can definitely see merits of fine-tuning the amino acid feed strategy. I hope that answers the question. I think I've got roundabout a little bit there.
All right. Our next question is, you targeted the initial concentration of AAs in the media in your AA replenishment strategy. Is it possible that the different target or targets might lead to better results? How do you know that the initial concentrations are ideal?
Yeah, it's a very good question. I mean, for this set of experiments, it wasn't fully arbitrary, but we did pick let's restore to baseline as our target to sort of prove that we could calculate depletion rates and react to them. We don't know, to be honest. I mean, we contemplated pyramid feeding amino acids as well relative to VCD. Again, we don't know whether that's going to have an effect. It's kind of opened up quite a lot of other ways that we can look at our process to further characterize and fine tune it. Because we've not really broken that down, as in adding different amino acids at different levels at different time points. It's a great question, and it's probably one to go back to the Amber 15 with to do a bit of a screen. What we wanted to show is that we could take a process all the way through from screening to bioreactor manufacturing relevant levels, and improve its performance or retain its performance throughout that scaling, which we have achieved with this project. But there's certainly more we can do in terms of fine tuning what to feed when. Do we want to feed different groups at different times, and why are we doing that? It would be going back to the screening, I think. But we certainly don't know if the baseline levels are ideal, particularly considering they're all quite similar across the different medias that we screened, and the performance was quite dramatically different from the high to the low. So media 6 to media 9 that we used. The titer was probably threefold lower with media 9, even though the baseline composition was very, very similar.
All right. Well, it looks like we're running a bit short on time. So if we don't get to your question, we'll be passing them along to Chris, who can follow up with you directly. So please go ahead and continue to type in any questions that you might have, and we'll get those passed along. The last question for the webcast is, were there any changes in N-glycans?
We didn't see any striking changes in N-glycans, but we hadn't looked at them in a great deal of detail until the latter stages. So we could go back and look in a bit more depth. So we needed to have enough volume to purify from to look at N-glycans, and the only scale we really were able to do that was at Amber 250. So we haven't seen any dramatic differences in N-glycan profile, in short, but we didn't look at, that was only in media 6, so the one media with some slightly different conditions. We might have seen differences maybe earlier in the process development, but we didn't have enough volume really at the end of the culture in the Amber 15 scale. But it's certainly something that we're really interested in looking at, and we did monitor at the latter stages of process development. We didn't see any dramatic differences, in short.
All right, fantastic. Well, thank you so much, Chris.
Thank you.
And thank you to our audience for joining us. The recorded version of this webcast will be made available for viewing on our on-demand website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.